First, we need to clean the aquarium to remove the leftover algae. This step is important because it prevents contamination during the experiments. A clean aquarium also ensures that only the microalgae are affected by the fertilizer, so this helps us to obtain reliable and accurate results.
Next, we prepared 30 ppt salt water using marine salt. The salt was measured and dissolved in clean fresh water until fully mixed. This salinity is suitable for microalgae culture and helps to ensure proper growth.
First, we set up the tanks to prepare suitable culture environment. Each tank was cleaned to remove any contamination, then filled with water at the required level. Aeration was added before inoculation to ensure enough oxygen for the culture. Proper tanks setup is important because it creates stable conditions and helps the organism grow well through the experiment. Next is randomization and labeling. Each treatment was assigned randomly to the tanks to avoid bias in the experiment. Every tank was clearly labeled according to its treatment, prevent confusion during observation and data collection. This step is very important because it ensures the results are fair, accurate, and reliable.
Next is the upscaling method. Microalgae were transferred from small culture to a larger culture volume. The algae were taken by a clean pipette and gently added into the larger tank. This process helps to increase biomass while avoiding contamination.
Next, we take salt water as the growth medium for microalgae. Salt water provides essential minerals and the correct salinity for the growth of microalgae. It also imitates the natural habitat of microalgae, and this also ensures optimal growth conditions throughout the experiments.
Now, we prepared and show all the treatments, which include control, NPK and F2. The control is used as a reference, while NPK and F2 are experimental treatments. These treatments allow us to compare their effects on the culture. By using different treatments, we can evaluate which method gives the best results.
Ready, NPK and F2 fertilizer were applied. A measured amount was added on the selected tank. This step was done to compare microalgae growth under different nutrient treatment.
Next, we use the hemocytometer to count the number of microalgae cells. These tools allow us to measure microalgae growth precisely. By counting the cells, we can observe changes in population over time. This helps determine the effectiveness of the fertilizer.
Next, spectrophotometer method. To validate our findings, we use spectrophotometer to measure absorbance at a specific wavelength. While cell counting tells us the population number, the spectrophotometer measures optical density, which represents the total concentration of biomass and chlorophyll pigments. By measuring how much light is absorbed, we can quantify the total suspended organic matter within the culture. Unlike manual counting, which can be prone to human error, absorbers account for cell volume and the accumulation of pigments. This serves as a reliable proxy for total biomass yield, ensuring our data reflects the biological density of the microalgae population beyond just the individual cell count. The growth performance of microalgae, we are using under 3 nutrient treatments, which is T1 100% NPK, T2 50% of NPK and 50% of F2, and T3 20% NPK and 80% of F2. We monitored 2 key indicators over 11 weeks: cell density to measure population count and absorbance to measure biomass concentration. Looking at the cell density graph, the results show highly dynamic growth with several peaks. The standout performer here was T3, which achieved the highest single peak of the study at week 5. This indicates that the 80% F2 ratio in T3 is excellent for triggering rapid cell division early in the culture cycle. Meanwhile, T1 shows its strength later in the experiment, reaching its maximum count at week 10. Next, we look at the absorbance graph. Interestingly, the highest population count in T3 did not result in the highest absorbance. Instead, T2, the 50-50 mix, recorded the highest overall absorbance value, peaking at week 10. This suggests that while T3 produced more cells, the cells in T2 were likely larger or contained higher concentrations of chlorophyll, leading to more dense biomass. In conclusion, T3 is the most effective for achieving a fast population bloom, but T2 is the superior treatment for maximizing total biomass yield and pigment quality. This result demonstrates that a balanced nutrient mix is a key to sustainable, high-quality algae growth.